start with chapter 9, again doing things so totally out of order, looking at microbial growth. Okay, so in this chapter, we're going to define what is growth for bacteria, we're going to talk about how you measure it, and we're going to focus a lot on the different influences on bacterial growth. Okay, so when measuring bacterial growth, okay, you do not measure it in the traditional setting of size. Okay, bacteria, they do not grow like we do. They do not mature like we do. They pretty much stay the same size in their tiny little lives. Okay, so that's not how we look at microbial growth. Instead, we look at microbial growth indirectly as population size. Right? How many cells are present over a certain amount of time? Okay, that's growth. You folks have already done this. Okay, looking at the cloudiness, the turbidity in test tubes, right? And I asked you, how cloudy is it? That's a measurement of growth, right? The cloudier it is, the more cells that are present. So why is it that we measure microbial growth and population size? Because of how they reproduce. Okay, first chapter we talked about binary fission. This process of essentially self-cloning, where one cell will duplicate itself, becoming two identical cells, barring any mutations. Now, a very straightforward process, way less work than like mitosis or meiosis, but okay, you start with one cell, and the cell copies its chromosome and some other components. They get segregated. A septum begins to form, pinching off the cell wall and membranes, and then, ta-da, you have two genetically identical cells. And side note, if they did not come apart, that's how arrangements form. Okay, strep chains, staph clusters, what have you. So one cell becomes two. Then those two duplicate, becoming 4. Then 8, then 16, then 32. This is a very predictable process, binary fission. Just copying themselves over and over and over and over again exponentially. It is so predictable that it's quite easy to measure the amount of time it takes to go through a single generation. So we call this doubling time, or sometimes books call it generation time. How long does it take for that population to double? From 1 to 2 or 8 to 16, whatever. So you might wonder, okay, how long does that take? Right? How long does it take for bacteria to go through a single generation? Not long. Okay, E. coli is around 20 minutes per generation. By the time we're done with class, that's nearly 3 generations that E. coli would have gone through. Some bacteria do get a bit faster. Vibrio species, for instance, like Vibrio cholerae, okay, causes cholera. Generation time around 10 minutes. Others, though, get very slow. Tuberculosis, for instance, those cells can take a couple of days to duplicate. Okay, so it all depends on the organism, but they tend to be quite quick in their doubling time. So just to kind of conceptualize this, make it more real, let's look at, and this chart's in like every book, but this is my favorite version because it's the easiest to understand. Let's look at a 4-hour period, 240 minutes. And in this 4-hour period, we're going to start with a population of 10 E. coli cells. So every 20 minutes, they double. They go through a round of binary fission. So 4 hours is actually 12 generations. So 10 becomes 20 cells, 20 becomes 40, 40 becomes 80, and look what happens after 4 hours. You've gone from 10 cells to 40,960. It's a decent amount of cells. Well, let's consider what happens if you let it go for 24 hours unchecked. Well, that's a slightly larger number. If you let E. coli go for 24 hours unchecked, it would number around 8 times 10 to the 29th cells. So 8 with 29 zeros. I have no idea what that number is, but it's big. That is such a large number that E. coli would basically consume the entire planet. But here's the thing, folks. I'd imagine that doesn't seem particularly realistic, right? This is beautiful on paper, but there are things that influence this. So let's start thinking about that now, because that's a big part of this chapter. What things do you think would prevent this from going so perfectly in any given environment? What are things that influence your behavior? Things that make you go faster or slower? Nutrients. Let's see. What else? What within the environment? What sort of factors would you consider? What? Temperature. How about pH? Oxygen availability. Water availability. Okay, so that's a big part of this chapter, is considering that, yes, bacteria could reproduce unchecked, take over the planet in a day, but the thing is, in a real world setting, there's so many limiting factors. So while this is beautiful on paper, it's not totally realistic. Okay, so keep these things in mind, because again, we're going to get into it more later on. All right, so when it comes to reproduction, bacteria tend to be cut and dry, but there are other forms of microbial growth out there. Okay, and of course, with bacteria, there's always going to be those species that don't want to reproduce like the others. Okay, so for instance, Streptomyces, we don't ever get to work with those. I have. They reproduce almost like fungi do, like molds. Let's see what else. Ricketsias reproduce in a somewhat unique way. Chlamydias reproduce almost in a similar fashion to viruses. So there's some oddballs out there. Now, one interesting example of an organism that reproduces similarly to bacteria is good old yeast. Okay, yeast are fungi. Okay, they are eukaryotes. And yeast undergo a process called budding. Similar to binary fission in bacteria, but not quite the same. So in budding, the yeast will produce clones of itself as small buds. And these smaller versions of itself grow. But once that bud reaches a critical size, it breaks off. But it's a clone of the mother cell. And if you ever get the chance to see these under the microscope, you can stain them and you can actually count the scars. You can count how many times they've reproduced recently. So similar to bacteria, but not quite the same. All right, so the next few slides, no textbook really knows where to fit them exactly. So we're going to deviate kind of sort of for just a little bit here. The next couple slides deal with microbial ecology. Okay, this would be asking yourself, in any given environment, what sort of bacteria are there? Okay, how are they interacting with each other? Are they playing nice? Are they fighting with each other? I mean, that's ecology. But here we're looking at the ecology of bacteria in any given environment. So, most environments on this planet have bacteria in them. And most environments will contain a mixture of species. It's not to say there aren't environments out there that are dominated by a single species, but most of them have a multitude of different bacteria kind of living together. Now, imagining that pond in your mind and all the bacteria in it, most of them are planktonic. So that means they're free living. They mind their own business. They're just kind of doing their own thing. Others, though, as we'll see here in the next couple slides, can come together and work together, forming what we call biofilms. But hang on to that thought. So when you talk about ecology, usually in general biology, with ecology, you always want to talk about, in any given ecosystem, what are the different organisms doing there? Right? Are they getting along with each other? Are they fighting with each other? What's happening? And in Gen Bio, they got great examples, like, looking at all the different plants in the forest and all the different mammals and how they get along, and they're easy to interpret examples. But the thing is that ecology applies to bacteria as well. So when you put a bunch of bacteria together in an environment, sometimes they cooperate with each other. They help each other out. Other times, they'll fight with each other. They'll compete. So let's talk about how they can cooperate, right? How can bacteria work together? Well, one way was through those biofilms, which I'm going to come back to here in just a minute. But another way is through a kind of sort of bacterial food web. Now, if you've had Gen Bio recently, you probably remember food webs, right? The idea that there's always a bigger fish. So at the bottom of the food web, you got producers, plants, grasses. Okay, grasses are eaten by secondary consumers, or primary consumers. I always mix it up, but it's eaten by some sort of herbivore. Okay, those herbivores are then food for carnivores, wolves, eagles, hawks, whatever. So bacteria have their own version of a food web. Here, it's a food web of chemistry. So for instance, at the top of the food web, say we have some type of macromolecule. Pick your favorite. Protein, lipids, DNA, who cares? Pick a macromolecule. Well, that's going to be food for bacterial species number 1, whatever it is. So it's going to gobble up that macromolecule and it's going to crap out some kind of waste. So it's going to release some organic waste, whatever it may be. Well, that organic waste can be food for a different species of bacteria. So say species number 2 is like, your poop looks delicious, I'm going to eat it. All right, so it breaks down organic waste into, say, carbon dioxide. Okay, is that the end of the road? No, because there's organisms that can fix CO2, like plants and other photosynthetic bacteria. So that's species number 3. He would handle that. And it just continues in kind of a cycle. So bacteria can work together to, in essence, kind of cycle through molecules, to start from something complex, turn into something simple, and then actually back to something complex. Oh, and then competition, right? You throw a bunch of different organisms together in every environment, any environment, they're going to fight. They're going to vie for real estate. They're going to fight for resources. Bacteria are no different. It's just they don't have horns or teeth or claws. They instead fight with biological warfare, with biochemistry. So the best example, antibiotics. Most folks don't realize that like 99% of antibiotics are produced naturally by bacteria. It's nothing we cooked up. We just found them doing it, and we took advantage of it. So bacteria will produce antibiotics to kill their competition. Okay, so that's a great example. And they do make a number of other nasty toxic substances to kill off competition. All right, so before I start talking about biofilms here, I use too much time doing this, but I like to talk about 3 hopefully relatable examples of food that is the result of microbial communities. Okay, so I like to see if folks have never seen these before. And I know you guys are so super quiet, but please help me out here. All right, so on the left, there's a jar of kind of a golden colored substance with a scary looking white mat of something on top. Anybody know what that brown liquid is? What if I were to tell you it's fermented tea and it tends to taste quite acidic? Hey, we got a winner. Yes, it is kombucha. Now, kombucha is all the rage right now. Tons of flavors, ranging in acidity, but folks don't realize how it's made. So this is one of the only things I've not made at home yet. I mean to. But when you make it at home, you steep tea, sometimes you throw in sugar, and then you have to put in that weird looking white mat. Okay, that's called a SCOBY. That's your word of the day. A SCOBY is a community of yeast and bacteria that work together to ferment that tea and whatever sugars are in there. All right, I've had now officially 2 people in the last year and a half that know what the top right image is. So in the palm of that person's hand, that is not cottage cheese. Okay, so it's not cottage cheese. Anybody ever seen those before? Usually, I've not seen a lot of folks who make this at home. It's actually really easy. Was it? No, it's not cheese at all. It's used in a dairy-based product that's very similar to yogurt, but a lot more liquidy. We got some kefirs over here. Yeah. So kefir is another popular beverage right now, right? Super probiotic. It's like drinking yogurt. Those are what's called kefir grains. Itty bitty balls of microbial communities. Okay, different set of bacteria and yeast from the kombucha there, but this one's really easy. You just got to find somebody with the grains, and you take some milk, you throw in the grains, you let it sit on the counter for a day or so, and once it gets a little viscous, you strain it off, and you're good to drink. It is not like the stuff in the store, though. It's like effervescent. All right, now bottom right there before we move on. That one's very near and dear to my heart because I love baking. There's your hint. Anybody happen to know what that is? Sourdough, right? Yeah, that is a sourdough starter. I have one. It's officially 7 years old because that's how old my son is. I made it like while he was home after the hospital. But unlike normal bread, which is just throwing in powdered yeast, this is a community of lactic acid bacteria and yeast living in harmony to make bread that's slightly acidic and actually has a considerably longer shelf life because of the acidity. But yeah, I just like to share those with you because we take for granted a lot of the things we consume are the product of microorganisms working together. All right, so enough of the fun stuff. So what you see in most of those images, excluding sourdough, is biofilms. Okay, so biofilms are basically microbial hotels. So some bacteria get together and they decide they want to cohabitate. They want to create housing for themselves. So what is that housing? It's sugar. It's a polysaccharide encased community. So think of it as this web-like goopy network. Like a spider web was much thicker and much goopier. Best way I can describe it. And the sugars that they produce in this tend to be fairly unique. All right, so let's look at the evolution of a biofilm. So biofilms form traditionally on surfaces. So you start with some planktonic bacteria, right? These guys are planktonic, and they're like, hey, let's make a biofilm. So how do they do that? They communicate with each other. Now, this is mostly a footnote here, but the process of communication is called quorum sensing, right? Like having a quorum when you have a committee together. Quorum sensing is actually fairly complicated stuff, but long story short, they just make chemicals that say, hey, come this way. So they signal to each other that they want to stick to this surface. Okay, so sticking to surfaces. Usually, we see this with bacteria that make slime layers or capsules, okay, glycocalyces. So they collect on the surface, they reproduce, and then they start secreting this orange stuff. Okay, the orange stuff is that polysaccharide, or more technically termed, extracellular polymeric substance, EPS, okay, sugar. So they start secreting and expanding, usually they're glycocalyces. So you end up with this goopy web-like network of sugar. As you can see, over time, the biofilm grows. And it can be quite dense, very, very thick. And it gets so large that you can get other bacteria joining in. Now, eventually, that biofilm gets so large that it can start breaking off, and the cells can go out, and they can start making new biofilms elsewhere, beginning the process again. Sticking to a surface, secreting this EPS, and creating that biofilm. So we bring up biofilms in this class mostly because of the fact that many pathogenic bacteria make them, and they make them inside of you. And the problem with biofilms is that they're very difficult to treat. They're so dense, antibiotics don't penetrate well. So look at kids with chronic ear infections, right? Little kid gets an ear infection, you get the pink amoxicillin, they take it, it seems to go away, and then 3 months later, boom, it's back. Well, in many cases, that amoxicillin kills out that biofilm to a point, and then it starts slowly coming back. And it becomes a vicious cycle of going through bottles of amoxicillin on and off. The other problem is that your white blood cells have a hard time of breaking down big old biofilms. Okay? It's just difficult for them. So it's an unfortunately common trait we see with many bacteria. All right, so putting into pictures here, the image on the left is the one you see in like every textbook. They try to show you that web-like EPS between the bacilli. I got this from a journal article, though, on the right. I like this one because I feel it gives you a better impression of the density of the EPS. Right? There's way more of that webbing between the cocci than you see in this image. So I need to trim this down. So I'm just going to cover a couple examples here. So biofilms, again, are pretty darn common. Okay, especially amongst disease-causing bacteria. So my favorite example to lean on is dental plaque. Right? Every day we brush our teeth. And not only are we scrubbing out food, we're also fighting an uphill battle against the biofilms that form on the surface of your teeth. And disclaimer, you don't have to know any of this. Okay, this is just for me to make this more relatable. Biofilms, or the biofilms that form on your teeth, are actually incredibly complex. Okay, so for example, love this picture. I mean, it could be better, but this was published by Scientific American a few years back. They did a wonderful piece on dental plaque and the biofilms that form on your teeth. But just to show you how they evolve. So after you eat, or after you brush rather, and eat, 2 species of bacteria will stick to your tooth. Okay, those in the genus Streptococcus. So Streptococcus mutans, Streptococcus salivarius. We all got it. There's no avoiding it. It sticks to your teeth as soon as it gets the chance. These guys start the biofilm. And over time, the biofilm grows. Apparently, it's got names for the different regions. But as it grows, some more unique bacteria can join in. And the longer the plaque goes, the yellower it gets, the nastier the bacteria. So, for example, in the anoxic zone here, no oxygen, you got these yellow lines, if you can see them. That's fusobacteria. Okay, they only join in once the plaque gets really old, and they are the best at breaking down enamel. So the longer this goes, the worse the bacteria are. The better they are at breaking down the enamel on your teeth. But there's way, way more to it than that. But just to show you how complicated these biofilms can get. We talked about persistent ear infections. Catheters are a big problem. Okay, catheters can encourage the growth of bacteria inside the body. Jumping down the bottom, because they can form on the insides of pipes or drainage, they can break down plumbing over time. Let's see what else. But they're not all bad. Okay, looking at wastewater treatment and bioremediation. Okay, we can use biofilms as natural filters to clean out the stuff that we put into our rivers and streams or what we flush away every day. So the bacteria in these biofilms can scrub it out. So yeah, I need to clean up this slide. Okay, so that's biofilms. But any questions about biofilms or what they are or how they form before we go on? All right. So this next part, what I want to transition into now is something called growth curves. Okay, so I want to talk about what you look for in terms of growth for any given species of bacteria. So let's say we go back to our proverbial, whatever. We go back to that pond I told you to imagine. And say you want to start isolating species of bacteria. How do you do that? How do you get a pure culture? Aseptic technique. Streak plating, right? Get isolated colonies, pick the colonies, and put them on their own plates. Right? This is nothing new. We've been doing this in lab. So what we don't get to do in lab, which is a shame, is this next experiment. So once you've isolated that new species of bacteria, one of the things we like to look at is their lifespan. What does their growth look like over a period of time? So what we want to plot here is called a growth curve. The very predictable lifespan of any given bacterial species. So to plot a growth curve, you have to set up a fairly simple experiment. Okay, most of what you need is in this picture. So we need some flasks. Could be 1, could be 20, depends on how many species you're testing. But regardless, you need some flasks, and in there, you have growth media. Pick your favorite. TSB, nutrient broth, whatever. You inoculate each one, close it off, and then the machine here, called a shaker, does what it does. It shakes the flasks. So you set this up, okay? Inoculate some media, shake it, and what do? You sit there and you stare at those flasks. And you plot cloudiness over time. That's the experiment, okay? Doing what we've done with broth tubes in the past, but in a more precise fashion. So the thing to consider as you do this, as you stare at these flasks that are shaking, you do not add or remove anything. So we call this a batch system. It's closed. So once those cells go in, that's it. You're not adding any new food. You're not taking out any waste. You're going to sit there, watch them slowly die over a period of time. Okay, that's the experiment. So while it is kind of gloomy that you're sentencing these poor cells to death, you do get valuable data from it. Okay, so growth curve data. So this is what you're trying to plot here. Okay, so let's look at the axes. Whoops. So it says the logarithm of living bacterial cells. Yes, technically that's very correct, but let's just do it like this. The number of cells, i.e., how cloudy is your flask, versus time. So again, how cloudy is your flask? How long have you been staring at it? That's what you're plotting. And what you're trying to plot here are the 4 phases of a microbe's lifespan, of a bacterium's lifespan. All right, so I'll say this now and I'll say this at the end of this part. It does not matter what species of bacteria you test, you always see the same 4 phases. Okay, so every time this starts, it starts with lag phase. Aptly named because not a lot happens. So in lag phase, you can see the slope really doesn't change, right? It's flat. The cells are adjusting to their new environment, right? You just threw them into this new scary environment, they got to get used to it. So they have to ramp themselves up metabolically. That's pretty boring. Now, all of a sudden, something exciting happens. If you think this is exciting, your flask becomes cloudier. And not only that, it's becoming cloudy rapidly. So here you can see the slope takes off steadily. So you call this log phase, or sometimes called exponential phase. So in log phase, this is where the cells are reproducing at their maximum rate, as fast as they can. And with that in mind, this is also where we calculate doubling time. Remember how I said E. coli takes 20 minutes to reproduce? It's calculated from log phase. The calculation is actually really easy. So some other things of note with log phase. This is where bacteria tend to be the most sensitive to antibiotics, because antibiotics inhibit growth. And 2, this is where we see the appearance and production of primary metabolites. So let's talk about what that means. Okay. All right, so a metabolite. A metabolite is anything that your cells can make. Anything a cell can make is a metabolite. So if your cells can make amino acids, metabolites. If you can make nucleotides, metabolites. Anything a cell can make on its own is a metabolite. So we're going to talk about primary and secondary metabolites. Primary metabolites are necessary, key term there, necessary for growth. So cells need amino acids. So they make them because they need them for growth. We need nucleotides to make DNA, right? Primary metabolites. Now, why I bring this up is because many of the supplements we take for granted that we buy from the pharmacy, many of the supplements are harvested from bacteria that make them naturally. Okay, so many primary metabolites can be of valuable use to humans. Okay, maybe it's something we can't make, but good old E. coli can. We just take advantage of them for their ability to do it. All right, so lag phase, not a lot happens. Log phase, they take off. And now the sadness begins. Stationary phase. Look at that poor slope. It's leveled off once again. What happened? This is a closed or batch system. Food is finite. And in stationary phase, they're starting to run out of food. And that's reflected in the growth. Less food means slower growth. On top of that, wastes are accumulating. Now, for some bacteria, wastes aren't necessarily too bad. Maybe carbon dioxide. For other organisms, though, waste can be all sorts of acids. Like poor E. coli makes 5 different acids from waste. It kills itself by stewing in its own acidic waste. But yeah, so now the growth has leveled off. Less food, more wastes means a bad environment. Now, in addition, we start seeing the production of secondary metabolites. So what's the distinction here? Well, primary metabolites are necessary for growth. Secondary metabolites are not. These are molecules produced in response to stress. So, classic example, antibiotics. Bacteria aren't going to be making antibiotics when they're happy. They don't need them for growth. But you bet they will make them when they're stressed out, when they think they're being attacked. Okay, and there's others too. Endospore formation molecules would be produced here. Those are necessary for stress, amongst others. But again, I bring it up because secondary metabolites can be of use to humans. We can take advantage of bacteria that produce them naturally. All right, and then the saddest phase of them all, death phase. Look at that poor slope. Now it's declining. So at this point, food is gone. Okay, some cells can cannibalize each other, but some can't. Okay, but food runs out. Wastes are through the roof. So now the number of new cells being produced is far outweighed by the number of cells dying. And you see that flask gradually lighten up. Now, the length of time this takes is highly variable. Okay, it depends on how fit or tough the organisms are. This could be a matter of hours or it could be a matter of years. It really depends on the fitness of the organism in question. So, those are the 4 phases of any given bacteria's life cycle. Lag phase, log phase, stationary, and death. Perform this experiment with any species of bacteria, you're going to get those 4 phases. What varies, though? The 2 axes. How cloudy did your flask become? How long were you sitting there staring at it? Okay, so for instance, and the reason why we can't do this, to do a growth curve for E. coli takes around 3 to 4 hours. Okay, we don't have that sort of lab time, but it does take some time. All right, but any questions about the 4 phases? This I threw in here from an older text because it's one of the only ones that shows where those primary and secondary metabolites are produced. But yeah, see, during log phase, that's where you get the primary metabolite production. And then stationary phase is where you get those secondary metabolites being produced. All right, so on that note, there is a way, instead of sentencing your cells to death, to keep them alive forever. Okay, so there is a way of maintaining continuous cultures. So to do that, this is something we don't have, and you'll see why, you need what's called a chemostat. So in the bottom right image here, that's a chemostat. Right? Check out all that plumbing, got the little propeller here. This is the opposite of a batch system. With a chemostat, you control everything. Food, waste, oxygen, pH, agitation, okay, temperature. Everything is under your control. So you can maintain a culture in log phase or stationary phase forever. So chemostats are very valuable because This is how we harvest many supplements and pharmaceuticals from bacteria that make them naturally. So, for instance, in the image in the top here, this is a biorefinery. Each of these is a giant chemostat. And they could be producing antibiotics here. They could be producing biofuels. Okay, we feed the organism and we filter out the effluent, the valuable products. So for example, our friend from lab, Bacillus subtilis. Most folks don't know this, but B sub is the workhorse for polymyxin B production. Okay, polymyxin B is a neosporin. But they grow it in these huge vats, trickling it food, stressing it out enough so it produces tons of polymyxin B, which we then scrub out. Oh, and why do we get to do these in lab? That one unit there, probably a 1 to 3 liter unit, about $25,000. We used to work with these, and like a lab would have one nice one and then like a bunch of homemade ones. We got really inventive. But anyways. All right, so that's chemostats. All right, so probably get through a few of these slides. But the next thing we're going to look at here, which basically most of the rest of the PowerPoint, is talking about the different environmental effects on growth. Right, going back to that slide where I said E. coli would take over the planet, but it doesn't because of those things we talked about. These are those things we talked about. Okay, we're going to talk about how temperature, pH, oxygen availability, and water availability influences where organisms grow. And unfortunately, there's going to be a fair amount of classifications coming up because of how bacteria are ubiquitous. So, while we can find bacteria and archaeans in just about every environment on this planet, as we go through all this, just keep in mind, most of them prefer the environments we do. Okay, room temperature, neutral pH, oxygen is present, not particularly salty. Okay, most of them like the environmental conditions we do, but as we've seen, because of the ubiquity of microorganisms, they'll occupy even the most extreme environments. Okay, so the first physical requirement I like to talk about is temperature. So folks, I would argue that every organism has a well-defined temperature range in which it will grow. Right? We have a maximum temperature we can tolerate. We have a minimum temperature we can tolerate. And somewhere in the middle is that sweet spot, that optimal temperature that we work best at. Okay, that's every organism. Every cell has that well-defined temperature range. Now, what influences that primarily, but not exclusive to, protein or enzyme function. Okay, and we'll talk about this more in the next chapter, but you make it too hot, proteins denature. You make it too cold, well, enzymes can't do what they do. But we'll talk about that more in the next chapter. All right, so depending on the book, there's somewhere between 4 and 5 different temperature classifications. OpenStax is one of those books that omits kind of a gray area, which I'll mention. But life persists on this planet somewhere between -5 Celsius, so below freezing, to 110, okay, boiling. So starting from cold to hot, with cold, there are bacteria that can live at sub-zero temperatures. We call them psychrophiles. Okay, obviously, phile is to love, right? Psychro means cold. So cold-loving microorganisms. So the range for these guys is somewhere between -5 Celsius, below freezing, to 15 degrees Celsius. We would find these organisms living at the bottom of the coldest parts of the ocean, where the pressure keeps it from freezing, but it's well below freezing. Or maybe locked under Antarctic or Arctic ice. Okay, these guys would be doing just fine. All right, now the gray area. Psychrotrophs. Poor psychrotrophs get left out of some books. But psychrotrophs occupy kind of a narrow band around here. Psychrotrophs are cold, tolerant bacteria. Some folks argue they're more mesophiles than anything, but these guys tend to do better at temperatures a little colder than room temp, maybe refrigerator temps. And that's exactly where we look for them. Think food spoilage, right? You leave anything in the fridge long enough, it's going to go bad eventually, right? It's going to get green and fuzzy, slimy, whatever. Psychrotrophs. All right, now mesophiles. That's us. We're mesophiles. 25 to 45 Celsius. So fairly cold room like this one up to a hot summer's day. So 25 to 45. This is most organisms, and this is, as far as we know, just about every pathogen. All right, thermophiles. Now we're getting hotter. 45 to 70 Celsius. Hot day to very hot shower. So here, these are the organisms you'd find in some hot springs. You find them in hot water heaters because, right, they maintain a constant hot temp. And compost heaps. And that would seem strange, but if you've ever been around really big compost heaps, the core gets extremely hot because you get thermophilic bacteria that generate a hot environment that are really good at breaking down those banana peels and coffee grounds you threw in there. Okay? They actually will steam if you turn them. All right, and the last one, and this was not in the books like 15 years ago, hyperthermophiles. I give credit to the folks who discovered these guys. 70 degrees Celsius to 110, right? We're at boiling now. Typically, these are archaeans, some bacteria, but these were discovered growing near the volcanic vents at the bottom of the ocean. Okay, and you'd think around this crazy hot volcanic vent there should be no life, but it's teeming. Okay, so you get bacteria that must have, or archaeans, that must have this extremely hot temperature to survive. Okay, so somebody, I give them credit, had to figure out, oh yeah, you need to grow them at this temp and they'll do okay. So that's the 5 classifications for those temperature ranges, from psychrophile to hyperthermophile. Now, a couple things to take home from this. Most importantly, with these temperature ranges, if you want to grow a psychrophile, say it has to grow at 0 degrees temperature, 0 degrees Celsius, can you grow it at room temp in one of our incubators? No, that'd be too damn hot. Okay, same thing with the hyperthermophiles. Say you want to grow those at room temp, is that going to happen? No, way too cold. Every organism must be grown in its specific temperature range. And then the last fun thing worth mentioning, even this brand new textbook is wrong in its temperature range. Latest research has shown the bottom limit is somewhere around minus 15 Celsius. And the upper limit, we've known for some time, is around 121 degrees Celsius. That record belongs to the very cleverly named Strain 121, an archaean we found growing in an autoclave. All right, let me bang these out real quick, just to get us in a good place. All right, so, and I'll rehash over the temperature stuff next time too, but with the next 3 classifications, you're going to see two terms come up, obligate and facultative. So obligate is like an obligation, right? If you're obligated to something, you got to do it. Obligate means you must have something. Okay? We are obligate to oxygen. We have to have it. Now, facultative, on the other hand, means more flexibility. Okay, maybe they can live with oxygen, or they can live without it. They just don't care. Okay, so we'll see these two come up in the next few examples. All right, and then we were looking at, and this is where I really want to pick up on, is oxygen requirements. Okay, because an assumption a lot of people make is that all organisms need oxygen to live. And that's so very far from the truth. Okay, that's most eukaryotes, right? All the organisms and animals we're used to seeing need oxygen. But with bacteria, there's diversity. Okay, for many bacteria, oxygen's toxic. So, first thing we considered was, how do they make ATP? We need oxygen to make ATP. We undergo aerobic respiration. So we'll see in the next chapter, some bacteria don't use oxygen to make ATP. That's anaerobic respiration. And then we'll cover fermentation in the next chapter as well, where you encounter those organisms that don't care. Okay, yay, oxygen's around, but oh well, I can live without it too. The other thing we discussed is you have to consider if the organism can deal with reactive forms of oxygen. A consequence of living near oxygen is that your cells will produce superoxide radicals. You can't help it. It just happens. Okay, and it looks harmless, right? It looks like oxygen, O2, with one little electron. This is an incredibly reactive and damaging molecule. Okay, it will ruin your memories. So being aerobic organisms, we got to be able to metabolize this, right, to break it down. And that's what enzymes are for. Now, there are two specific enzymes required to get rid of this molecule. So the first one is superoxide dismutase. And I know it's an enzyme because it ends in ase. It's one of the ultimate cheats in biology. If you know it ends in ase, it's an enzyme. So that enzyme, which we produce, takes superoxide radical and converts it into hydrogen peroxide. Now, as I mentioned last time, hydrogen peroxide is still pretty bad. So the next step is catalase. Catalase takes that hydrogen peroxide and converts it into something a little bit more reasonable, oxygen and water. So from horrible to slightly less horrible to, okay, I can live with that. So considering these two criteria, okay, how do they make ATP and do they have these enzymes? You end up with 5 different classifications. So obligate aerobes are like humans. We need oxygen to survive. We can't live without it. And we produce both of those enzymes. We have to, right? We live in the presence of oxygen. Now, what you haven't encountered in the past, most likely, are good old obligate anaerobes. These are a stinky group of bacteria. They don't use oxygen to make ATP. They use other things, as we'll see in the next chapter. And a consequence of them not using oxygen is that they can't stand it. Oxygen's toxic to them because they lack both of those enzymes. If they grow near oxygen, superoxide radical accumulates and they die. Now, the toughest buggers are the facultative anaerobes. Okay, now facultative, right? Flexible. Anaerobe is a bit of a misnomer here. Most facultative anaerobes actually prefer oxygen. They do better when it's around. But when it's absent, they don't just curl up and die. Not like the obligate aerobes. Facultative anaerobes survive via fermentation. Okay, it's a slower lifestyle, but they can live without oxygen as well. Now, because most of them prefer oxygen, most of them will produce both catalase and superoxide dismutase because they have to tolerate that oxygen that they prefer. All right, so these are the three most common. Now for the oddballs. Aerotolerant anaerobes are something we will not work with in lab. Okay, these are not particularly common. And it's all in the name, aerotolerant. They tolerate oxygen, right? Anaerobe, but they don't use it. So evolution's a funny thing. These are organisms that don't use oxygen to make ATP, but they do make those enzymes. Now, one we will get to see in lab, thanks to Heidi's efforts, is a good old microaerophile. All in the name. Micro, little bit of something, aerophile, oxygen loving. So, these guys love a little bit of oxygen. They can't handle high concentrations. That's kind of strange, right? So why is that? They will generally lack one of those two enzymes we talked about. Okay, they could have catalase and lack the first, or vice versa. By lacking one of those two enzymes, they're not going to tolerate oxygen in higher concentrations. Okay. So in lab next week, you will test for this. And this is the very same experiment that you will set up. This is what's called the thioglycolate test. So in thioglycolate media, and I'll be brief because we're just going to jump into this in lab next week. The tubes you'll be working with are mostly anaerobic. Sorry if I drew this a little too small. So O2. And then everything below, no O2. So in these tubes, just the very surface is aerobic. Everything below is anaerobic. So, very simple experiment. You're going to come in, we'll talk about it, and then you're going to carefully inoculate 4 different thioglycolates. Throw in the incubator, we come back on Wednesday, we take a look, and you just look for the patterns of growth. Okay? Where is the growth in relation to the oxygen? So obligate aerobe, right? Must have oxygen to survive, so you'd assume it'd only grow at the surface, right? Okay, so that would indicate an obligate aerobe. Now the obligate anaerobes, they don't do a good job here of representing the real growth. And as we talked about, these guys don't like oxygen. But the image is misleading. What I typically see is something more like this. Because I'm not going to draw a bunch of little red circles, sorry. They will grow up to the very edge of the oxygen, like they're challenging it. But they will not grow in the oxygen. Tube C, facultative anaerobes. Okay, so here you have growth all throughout. Heavy at the top, that just looks terrible. Heavy at the top, lighter in the bottom portion, but still growth everywhere. So this is showing you that they prefer oxygen. They love it. They grow better at the top, but they still grow in its absence, just slower. Aerotolerant anaerobes, as I've been told, because I've not really worked with these, you just get light growth top to bottom. Okay? They can grow in the presence of oxygen, but they don't mind it. Microaerophiles, and if your tubes behave, they will look like this, but that's a big if. Here you get this little band of growth here at the bottom of the oxygen. Okay, so they're not all the way at the top, they're not all the way at the bottom. They're kind of hovering in between. So they need a little bit of oxygen, but they can't handle a lot of it, right? They can't grow all the way at the surface. So with the exception of tube D, you should get good examples of A, B, and E next week. Okay, so you will get to see this with your own eyes. Any questions about the oxygen requirements before we move on here? Okay. All right, so pH is really, really easy and short to discuss. So pH is a lot like temperature. Okay, there's a maximum pH that we can tolerate. There's a minimum pH we can tolerate, right? Too acidic, proteins denature. Too alkaline, proteins denature. And then there's that sweet spot in between, that optimal pH. So much like temperature here. And only 3 categories here. Yay! So these are pretty obviously named, right? Neutrophile. Bet you can guess what pH they prefer, right? Neutral. This is all pathogens. This is most organisms. And my only fun example is Helicobacter pylori. Helicobacter pylori is notorious because it causes peptic ulcers in the stomach. Okay, so what happens is you get a fissure in the lining of your stomach, and then this organism colonizes the fissure, creating a nasty ulcer. So here's the thing, though. Your stomach is particularly acidic. This organism does quite well in your stomach, but I'm saying it's a neutrophile. It actually creates a neutral environment within the ulcer. They are really, really good at neutralizing the surrounding environment. And we've put a lot of research into this guy lately. Guy named Marty Blazer is like the foremost expert on it. I've seen him talk. He's a cool guy. A lot of his work is showing that half of the world population naturally has it in the mucosal lining of your stomach. It may play a role in controlling appetite and hunger with ghrelin production. You can control that. And there's been a few papers that are looking at Helicobacter causing colic in infants. Okay, if you've ever been around a colicky baby, I'm sorry. If you ever have to deal with one, I'm sorry. But there's no good pathology for colic. You got a 3 to 8 month old that just won't stop crying. If they're awake, they're screaming in your face. If they're asleep, you are thanking whatever deity it is you worship, okay? We don't understand why it happens, but there's been a number of papers that have found elevated levels of Helicobacter in the stomachs of colicky babies, so it might be causing some gastric distress. So a very interesting organism. All right, so acidophiles, name implies it, right? They prefer acidic pHs. What we got here? Below 5.5, down to actually near 0. Many bacteria are acidophiles, and there are a number of archaeans that will grow near 0. Alkaphiles, right? They like alkaline pHs. Find these guys in soda lakes or alkaline lakes. Some bacteria, but mostly archaeans. Now, some books argue that soil microbes are alkaphiles, and honestly, that holds true around here. Good Midwestern topsoil is fairly alkaline, around pH of 8. But the problem is not all soil is alkaline. Okay, go out the Atlantic coast, hit the Carolinas, where you get cypress marshes. The pH of those is like 5. Okay, that's acidic. So when you read that, take it with a grain of salt. All right, so that's considering pH. Water availability, and this one's kind of weird. When they talk about water availability, really what they're talking about is salt tolerance. Because we have to revisit osmosis. Because like we talked about last chapter, bacteria can't just drink water. They are reliant on osmosis for water to get in or out of the cell. Now, environments will vary in how salty they are. And it can be too salty or too distilled for the cell, and that can kill them. So that's what we're looking at here is how do they handle salty conditions or different levels of tonicity? So we're going to talk about halophiles. Okay? Bacteria that tolerate salts. So halo means salt. So in the last chapter we saw these. So there's 3 types of tonicity. In an isotonic environment, this is ideal. Both sides of the cell have the same amount of salts. Okay, so water can freely pass through. The cell's going to get what it needs. This is what it wants. Isotonic is ideal. Now in a hypotonic environment, slightly less ideal because the inside of the cell is saltier. So water's going to rush in. Now, here's the thing. They show this poor cell is blowed out, right? The cell wall has ruptured. Talk about this a bit more in the next slide or two, but gram-positives tend to be very resistant to these more dilute environments. Okay? They have a thicker cell wall, so they're less likely to explode. Okay? Gram-negatives, on the other hand, because of their thinner cell wall, don't handle these differences in salt well. So this is probably a gram-negative cell here. Okay, so hypotonic, more salt within the cell. Now the least ideal environment is hypertonic, where you have thrown your cell into a particularly salty solution. So as you can see here, water leaves the cell. Oh, I'm sorry. I meant to delete this slide because I managed to fit it here. Bonus slide, sorry. Okay, so this is what I want to get to. So what happens in that hypertonic environment? Okay, water leaves the cell, but look at the consequence. The membrane shrinks away from the cell wall, but the cell wall stays intact. This phenomena is called plasmolysis. Now, and I mentioned this, I think, in the last chapter. If you've had Fizz, you've done a similar experiment with red blood cells. And Terry or Sandy or Chris or whoever said, well, that's called crenation. This is not crenation. Crenation applies to cells that don't have a cell wall. Bacteria have cell walls, right? Plant cells would also undergo plasmolysis because plant cells have cell walls. Okay, I get people who confuse the two. Just want to mention that. So, for many bacteria, this just spells death. Water gets ripped out of the cell, poor little thing turns into a shriveled up trapped raisin. So how do you get around that? Well, some cells can adjust their internal solute concentration. They can start making things, making more solutes to compensate for the difference. So what I'm saying is, they're trying to go from hypertonic to an artificial isotonic environment. Okay, they're trying to adjust the internal tonicity. So this is why we use salting to preserve a lot of foods. That's why beef jerky is not going to go bad anytime soon. Okay, salt rips the water out of living cells. Okay, so with this stuff in mind, with tonicity in mind, there's a whole world of different bodies of water on this planet. And they all vary in their salt concentrations, but they're all teeming with bacteria. So obviously bacteria are going to vary in their tolerances of salts. So, 3 kinds of halophiles. So at the top, facultative halophiles. Okay, this is a very, very broad group, and it says up to 10% salts. That really only applies to gram-positive bacteria. So folks, if you had lab this morning, we mentioned this a bit with that mannitol salt agar plate. Okay, folks, at 2 o'clock lab, one of our plates will be super salty, where only gram-positives grow. So gram-positive bacteria tolerate salt just fine. Gram-negative bacteria tend to be more sensitive. Okay, they don't deal with it well. They, for the most part, can handle maybe 1.5% salt. All right, so next up we have obligate halophiles. So this would be the bacteria that you find growing in our oceans. Okay, they need a salty environment. Okay, so they have to have at least 3% salt to grow. If you tried to grow them on our Petri plates, they would die. It's way too hypotonic. There's not enough salt in that plate, and they may explode. And then a newer classification, which I wasn't familiar with until like 10 years ago, extreme halophiles. I've only ever worked with one example of this, Halobacter. But these organisms require at least 9% salt. So to illustrate how salty that is, when I used to help Pat make the plates for this experiment at NIU, because in their version of our class, they have a lab where they do all this testing. They do temperature testing, pH, and salt. We don't do it here for whatever reasons. But we had to make plates with 12% salt. And I will tell you, we used to throw away half of them because the salt would crystallize. It was so concentrated. So yeah, at 9% salt, that's obscenely salty. Few organisms can tolerate that. But the Halobacter, they loved it. And be glad you don't work with it because they smell terrible. Outside of that, there's archaeans that we found not long ago growing at the bottom of the Dead Sea. And that's sitting at 33% salt the last time I checked. There's a cool experiment somebody did a couple of years back where they put a dress into the Dead Sea, like anchored on some wood. And when they pulled it up after a year or two, it looked like rock candy. It was just covered with salt. So yeah. And if you ever go there, don't go in there with any open wounds. You'll be sad. Side note. But anyways, okay, so that's the halophiles. Most bacteria are just facultative. They can tolerate up to a fairly high level of salt, even though they may not prefer it. Others are obligate to salty environments, and others have to have extremely salty environments. But any questions with the halophiles? Okay. All right, so now we're going to transition from environmental factors on growth to nutritional. And I handle this a bit differently than a lot of the texts do. A lot of them throw this into the beginning of the metabolism chapter, which is what we're doing next anyways. But besides considering the different environmental needs of your organism, you also have to consider what to feed it. Okay, so bacteria vary greatly in what sort of nutritional factors have to be considered. All right, so first thing to consider, major elements. Okay, what elements does an organism need to grow? Okay, here we come back to SNOP. Remember, sulfur, carbon, hydrogen, nitrogen, oxygen, phosphorus. Right, every cell needs those elements. And there's some others too. Need potassium, magnesium, calcium, and iron. You don't have to know that list. Okay, all I want you to know from the major elements is the carbon source. Okay, that's the most important thing because look at us. We are organic from head to toe. So it's obviously important to know where does the carbon come from to make you. So two classifications here. So carbon can come from two places for any given organism. So heterotrophs. We're heterotrophs. We get our carbon from organic sources. Okay, from your food, from proteins, from fats, from polysaccharides. With bacteria, though, anything organic is fair game. There's bacteria out there to break down everything. Now, autotrophs, though, are more picky in their carbon source. Autotrophs only get their carbon from one place, carbon dioxide. That's it. Right? You go from heterotrophs where anything organic is fair game to autotrophs that just want CO2. Okay, so like plants. Plants are autotrophs. many bacteria are autotrophic. Now, if you've ever talked about plants in the past and photosynthesis, technically it's only CO2 for a little while. Plants turn CO2 back into sugar. That's why potatoes are so delicious, because they take the CO2 from the air and they turn it into starch. And then when making media, you also have to consider trace elements. Again, you don't have to know this list. Just know that there are other elements that have to be thrown in as needed. Okay, so sometimes organisms need cobalt or molybdenum. Marine organisms need strontium. Okay, this is the optional list. But again, what we care about, first and foremost, is carbon source. Okay, is it from organic or CO2? All right, so the next nutritional factor to consider is growth factors. So here, take your carbon source, whatever it is, CO2 or a Subway sandwich, who cares? Okay, you take your carbon source, what can you turn it into? Okay, that's growth factors, metabolites, the things your cells need to grow. So there are 20 amino acids. We saw that terrible chart, right? Amino acids are growth factors. Okay, you need 4 nucleotides for DNA. Nucleotides are growth factors. You got vitamins B6, B12, K. The list goes on. Okay, vitamins are growth factors. So why do we care? Why are we talking about these things? Organisms vary in their ability to make growth factors on their own. So for instance, the rarest of the rare are the prototrophs. Prototrophs are totally self-sufficient. You give them their carbon source, they're good to go. They don't need anything else added. They can make everything on their own. I have only ever worked with one. It's called Bacillus fastidiosus. Easiest media in the world to make. It was water, agar, and some salt, because it took the CO2 from the air and what it needed from the salt to grow. Easiest thing. Now, they're rare. What's more common in nature, we are these, is auxotrophs. Okay, auxotrophs can't make all their growth factors from the carbon source. They need help. They need supplementation. So as humans, I believe we can only make 11 of the 20 amino acids. Where do the other 9 come from? Your food, or from the bacteria that pay rent in your stomach. But you can only make about half of the amino acids on your own. You're an auxotroph. Okay, so organisms vary wildly in how auxotrophic they are. And this is what we call fastidious. So how fastidious are you? How picky are you? How much supplementation do you need? Some bacteria, like E. coli, is like one step from being a prototroph. E. coli can almost make all of its growth factors. It's barely auxotrophic. It's not very fastidious. But on the opposite end of the spectrum, you got lactobacillus. Lactobacillus suck to grow. To make media for them, you need this huge grocery list of ingredients, because they have adapted to dairy environments, right? Milk is super nutrient rich. They've grown lazy over time. They're like, all right, everything's here. I don't need to know how to make this anymore. So they're extremely fastidious. Okay, so bacteria vary wildly in how fastidious they are and how auxotrophic they are. So then, most bacteria are auxotrophs, very few are prototrophs. Oops. All right, third thing here. So we've got our carbon source. Okay, you take your carbon source, you turn it into those growth factors, if you can make them. But that begs one important question. Where are you getting the fuel to do it? Right? You just can't make things spontaneously. You need energy to fuel your metabolism. Energy is going to come from 2 overall sources for all living things. Right? At the beginning of it all, energy begins with the sun. Okay, photosynthesis. And when I say photosynthesis, most folks think plants. But bacteria were undergoing photosynthesis before the first plants arose. Okay, so there are many photosynthetic bacteria that derive energy from the sun to fuel their metabolism. And then chemotrophs. Chemotrophs are a very heterogeneous group. We're chemotrophs. We get our energy from chemicals, from the organic food that we eat. But with bacteria, with their diversity, anything organic, or for the most part, inorganic, is fair game. So while tons of bacteria get energy from things like sugar, there are others that can use pieces of metal and strip away energy from that. Okay, so chemotrophs get their energy from chemical reactions, from other molecules. All right, so putting this all together, there's going to be two ways I'm going to address this. So looking at nutritional diversity as a whole, all living things fall into one of four categories. So with the dichotomous key approach, okay, all organisms have some energy source. Okay, is that from chemical reactions, so chemotrophs, or is it from sunlight, phototrophs? Right? Because if you're alive, you get your energy from one of those two sources. Okay, go down to the next level in the dichotomous key. Once you know your energy source, what's your carbon source? Okay, is it from organic molecules like us? Are we chemoheterotrophs? Or is it from CO2? Are you a chemoautotroph? Okay, same thing with the phototrophs. There are photoheterotrophs, and then there's photoautotrophs. So if the dichotomous key doesn't work for you, there's another way I was taught that I also like to kind of piece this together. Make sure I got enough colors here. All right, so here's the game. Two categories, right? Energy source, carbon source. All right, so if energy comes from the sun, you're a phototroph. If energy comes from chemical reactions, you're a chemotroph. Okay, look at carbon source, right? Two sources. If you get your carbon from carbon dioxide, you're an autotroph. If you get your carbon from anything organic, you are a heterotroph. Okay, so here's my game. Okay, it's just mix and match. So you could have photoautotrophs. Energy from the sun, carbon from carbon dioxide. You can have photoheterotrophs. Now these guys are weird. Energy from the sun, but carbon from organic sources. That basically breaks everything you may have ever learned about photosynthesis. Okay, we see these guys as purple non-sulfur or green non-sulfur bacteria. They can do this. All right, so that leaves us with two more categories, right? You can have chemoautotrophs. Okay, those guys are the ugliest to learn about. Energy from inorganic sources like metal, carbon from carbon dioxide. So chemoautotroph. Sorry, the black marker is terrible. Somebody abuses the tip on it. Black. Anyways, so that leaves the last one. Humans. Chemoheterotrophs. Right? We eat our organic food. You strip it of its carbon and its energy. Okay? Many bacteria are chemoheterotrophs like us. They get their carbon and energy from the same source. So that's my little game. That's how I always kind of pieced it together. So, energy source in one column, carbon source in the other. So, photoautotrophs, energy from the sun, carbon from carbon dioxide, right? Photoheterotroph, energy from the sun, carbon from organic sources. Chemoautotroph, right? You get it. Any questions with how either of these approaches work? Dichotomous keys are great for some folks, but I just like it this way. It makes more sense to me this way. Okay, so again, if you're alive, you're in one of those four categories. So, in the future, this makes for fabulous matching questions, right? So, heads up there. I didn't say that. Oh God, I just recorded it. Anyways, so I just imagine people listen to this at home and they hopefully chuckle again. But anyways, so why are we talking about all of this? Why are we talking about these environmental impacts on growth, the nutritional impacts on growth? One simple truth. We all have taken the bacteria we work with in lab for granted. Right? They're on the slant, they're ready to go. You throw them on TSA, they grow. But the thing is, you got to know all that stuff before you can get a new species to grow. What's its temperature range? How does it handle oxygen? What's its pH range? What sort of salty conditions does it require? What's its carbon source? What growth factors does it need? What's its energy source? That's 7 different criteria that you have to know exactly before you can get a new organism to grow. And this is why we will never be able to culture like 99.999% of all bacteria that are out there. We have no clue what they need. Okay? You know, consider those archaeans that grow at the bottom of the ocean by volcanic vents. Who the hell thought, hey, let me make a petri plate at 110 degrees Celsius and see if something grows. Right? It's crazy. But anyways, okay, we've talked about culture media. I really should omit this slide now. But yeah, you throw together the ingredients that the organism needs. You add agar if you want it solid. You leave it out if you want it liquid. So something we mentioned in the first lab of the semester is that media can be either complex or defined. Okay, TSA, TSB, that's complex media. Contains many different nutritious sources of food. Beef extract, peptone, tryptone, soy extract. It's like throwing the proverbial buffet at the bacteria. Right? You go to the buffet, you're not going to eat everything, but you're going to find something. Okay, that's complex media. You put it together in the hopes that everything will grow. Now, defined media, we haven't really worked with yet, but we'll get a couple chances. With defined media, you have a more specific list of ingredients. You want to start dictating what grows. So maybe you're only adding glucose as a source of food. Or maybe you don't add any carbon source and you assume they use CO2. So you define, you control what grows. So just to compare, and please don't memorize these ingredients. Okay, that's what catalogs are for. So at the top, that's what would go in nutrient agar. It's basically TSA. It's very similar. So you got peptone and beef extract. 2 very nutrient-rich sources of protein and carbon. You got some salt in there, agar, and water. Okay, so there's tons of different carbon sources in there for the bacteria. Now, at the bottom there, that is the ingredient list for glucose broth. And if you're ever making glucose broth, you're trying to grow E. coli. Okay, because few other bacteria will grow on this. Only source of carbon is glucose. That's it. So either they can grow on that or they can't. And aside from that, there's really not a lot of supplementation thrown in there. there's a handful of salts. Okay, so E. coli, which is almost prototropic, will grow on this, but most other bacteria won't. Just doesn't contain everything they need to grow. So just to compare and contrast those. All right, so any questions with the nutritional factors or the two weirdo slides here I had on media? Yes?
You said the growth factors are like the primary metabolites?
Yes, growth factors are primary metabolites. All right, so let's see how much of this I can get done. I may record just a hair of it for you to listen to over the weekend. But the last thing I want to talk about here is how do you actually measure bacterial growth, right? How do you do a growth curve? There's a number of different ways. Overall, though, you either do direct measurements, where you're counting cells directly in some way, or, and this is way more common, you do indirect measurements, where you look at things like turbidity or weights. All right, so couple of examples of direct measurements. The first one is direct cell counts. And this is exactly what it sounds like. It is counting cells, one by one. This is useful if you want to know absolute total number of cells, but there's one big problem. Okay, and imagine counting cells under a microscope. Do you know if they're alive or dead? Not really. Right? So you don't know living from dead here. That's the big disadvantage. Oh, I forgot I changed this. So to do a direct count under the microscope, you need one of these expensive slides. And you apply some liquid culture right on this spot here. Because under the microscope, there is microscopic grids etched into it. So look at their blown up example here. Okay, you see all the little cells here in red in the grid? So this is what you do. And somebody left one of these here last year. I think they took it. It used to be a hand counter here. You sit there with a hand counter and you just go click, 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 click for every cell that you count in the grids. I've never had to do this, but I have friends who had, and they just sit there for hours, click, 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 click. That's the cheaper method. If you go to a nice university, and most of them have at least one or a few of these, you can bust out what's called a Coulter counter, or flow cytometer. You don't have to know that name. What this is a machine that does the counting for you. So you take that broth culture and you put it in a capillary tube, really thin tube. And the cells pass by some electronic detector. From what I've heard in talks, this is usually a laser. Every time the cell passes the laser, it counts it. Coolest one of these I ever saw, and this guy got hired because of how awesome his presentation was, Wes Swingley over at NIU. He started right when I was leaving. He worked with NASA engineers to make one of these where every time it counted a cell, it got shot into its own little chamber and captured. So they can look at the genomes of each individual cell in the population one at a time. But that's the kind of stuff you can do with NASA money. I think it came in there with like $1 million grant, something obscene. Okay, so that's great if you want to know total number of cells, but again, you don't know if they're alive or dead. So I'm going to do this next one here, and then I'll probably record the rest for the weekend. There's not much. So what if you want to know living cell counts? What if you want to exclude the dead cells? Well, I'm going to show you two ways to do this. The most common way is through what's called plate counts, or, as I learned it, viable plate counts. Okay, viable meaning alive, right? So you'll get the chance to do this in the last week of the semester. Okay, so this is the method you use for quantifying living cells. Because the important thing to keep in mind here is that living cells make colonies. And this is an experiment where you count colonies. So let's just look at the experiment here. All right, so the way this works is you start with some source of bacteria. For you folks in the last week, that's going to be some food item. Could be lettuce, could be green beans, could be strawberries, could be beef. Okay, you're going to take some source of food and then you're going to dilute it over and over and over again. Okay, so you set up what's called a, where is it? Serial dilution. Okay, where your original inoculum gets diluted over and over and over again. So in the example, they have it set up so that each dilution is only tenfold. So first tube is 1 to 10. Next one's 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000. Okay, so it's just tenfold more dilute with each transfer. So great. You set up your dilutions. What's next? You have to plate them. Did I get that side? Yeah. So there's 2 methods to plating. You'll get a chance to do both of these this semester. So you're going to take your dilutions and either do what's called pour plating, where you mix your sample with molten agar. So you get to make your own Petri plates. That's neat. So that's what you'll do with the food lab. Or with a different lab, what you can do is you can spread out your sample on the surface of the plate evenly. But in the end, in both cases, you want colonies. So you transfer the dilutions, you plate them, and then you incubate them. Come back a day or 2 later, and this is what you may see. Okay, plates with colonies. Now notice, on this end, where it's not very dilute, you have tons of colonies. But as it becomes more dilute, you get less and less colonies. So, there's a rule behind which plates you can count. So here it is. A countable plate has between 30 and 300 colonies. So let's look at these plates going across. So 1 to 10. Too numerous to count, right? Who wants to count that mess? There's just too many. Okay, 1 to 100. Same problem. Too numerous. Third plate. Have to read, yep. 54 colonies. Ooh, that one's countable, right? It's in that zone. 1 to 10,000. I counted this actually just to make sure. There should be, is there 17 there? Thereabouts. We'll just say there's 17. It's been a while. Too few to count. Okay, that is not statistically valid if you're curious. And then obviously that one on the far right there is also too few to count. It's only 3. So 54 colonies. What do you do with that? Well, here's the thing. You're counting living cells. And can a dead cell make a colony? No, it's dead. So only living cells make colonies. And every colony starts as 1 cell. So each of those colonies we call colony-forming units because they started as 1 cell. So you have 54 colonies. You had 54 cells to start. So what do? You multiply the colonies by the dilution factor. Okay, so 54 colonies on the 1 to 1,000 plate. So 54 times 1,000. 54,000 living cells were transferred. Okay, so that would tell you how many living cells were on that food item to start. It's a very humbling lab because most folks find out there's way more bacteria on your food than you ever considered.
All right, so another take on the viable plate count is through membrane filtration. So with viable plate counts, you're assuming you start with a source of bacteria that has a lot of cells, and you need to dilute it. With membrane filtration, here you want to count the number of viable cells if you're starting with an environment that's very dilute or has few bacterial cells, but you still want to count them. So in order to do that, instead of diluting your sample, you need to concentrate it. So that's where filtration comes in. So in the diagram, you can see that there's what looks like water. So imagine it's a river water sample. And you pull that water through a very fine filter called a membrane filter. Now you can see in this image here, the cells are larger than the holes on the filter. So as the water passes through that membrane filter, the cells concentrate on the surface of the filter. So after you've pulled your water through, here's the neat part. You peel off that filter and then you lay it on a Petri plate. And the filters are so thin that colonies will form on the filter placed on the growth medium. So in the image, that's an EMB plate, just like from lab. And all those green metallic colonies that you see there, that's probably E. coli. So if this was a lake water sample or a river water sample and you found all those little green metallic colonies, that would tell you that there's a lot of living E. coli and probably a sewage leak nearby. Okay, so here we're doing viable plate counts, but you have to concentrate a dilute sample instead of serial diluting it. All right, so two forms of indirect measurement. So instead of counting the sheer number of cells, here we want to attribute cell mass indirectly to cell numbers. Okay, so how do you look at cell mass? Well, the most common approach to measurement, hands down, is turbidity measurements. Okay, this is what most folks do when measuring bacterial growth. They look at cloudiness. Because imagine a cloudy broth culture tube. You know, the cloudier it is, the more cells that are present in that population. But there is a formal way of measuring that cloudiness. Okay, there is a tool to do it, and it's called a spectrophotometer. All right, so spectrophotometers are nothing fancy. Okay, it's a enclosed box with a light bulb. Okay, so here's your light bulb. The light would shine through a specimen. Okay, here it's just a blank test tube, but light is shining through it. On the other end of the spectrophotometer, you have a light detector. Okay, so that light detector is telling you two things. One is transmission. Okay, how much of the light got through the sample? Here it says, well, 100%. So all of it. Now, what's actually more valuable to research is absorbance. How much light didn't get through? How much was absorbed by your sample? So let's say we have a bacterial broth culture, and we want to know how cloudy is it? Okay, how grown up is it? So you take that bacterial sample, you put it into this spectrophotometer. The light shines at it, but as you can see, not all of the light gets through. Some of it's absorbed by the bacteria. So you want to know, okay, how much light got through, how much was absorbed. So in this example, okay, it says 20% of the light got through. Okay, not much. But we don't really care about that. We care about absorbance. So here it says the absorbance was 0.70. So we say that is a OD of 0.70. Okay, so where did I just pull OD from? It's what we call optical density. Okay, it's an inverse measurement of the cell density. Okay, so here, as the culture gets cloudier and cloudier and cloudier, more of it gets absorbed and the optical density goes up. So this is what typically gets published in journals. And folks, remember the growth curves where you're plotting cloudiness over time with lag, log, stationary depth? This is how you do it. You sit there measuring the cloudiness with a spectrophotometer over however much time. Okay, but the disadvantage here, you have to have a cloudy sample. It has to be pretty heavily grown. All right, now, the last form of measurement is total cell weight. And this is exactly what it sounds like. It's weighing cells. Now, this is not commonly done with bacteria anymore. This I see more typically with my mycology friends, okay? Folks who work with fungi or molds, because this is pretty much the best way to measure the growth. So, to get the total cell weight of, say, bacterial cells, you need a centrifuge. Okay, so in the image, those tubes have been centrifuged. And what you see is what's called a pellet. Okay, so these whitish looking blobs at the bottom, those are the pellets that are left over after these tubes have been spun really fast. Okay, so the pellets are all the bacteria concentrated together. So you can get wet weight, where you weigh that wet pellet, or you can get dry weight, where you throw it into an oven for a little while and measure the dry mass that's left behind. Another technique that's used, and this is a fun fact, sometimes we freeze dry bacteria. Okay, and to do that, you use what's called a lyophilizer. There's your word of the day. I mention it because it is actually commonly done with fungi, to freeze dry them and then weigh them. But again, not commonly used. All right, folks, that is it for Chapter 9. If you have any questions, please let me know.